Assalamualaikum and hello everyone. Today we're going to look at Chapter 9, Reproduction and Development. So there are 4 subtopics in this chapter, which are 9.1 Sexual Reproduction in Flowering Plants, 9.2 Human Reproductive System, 9.3 Fertilization and Fetal Development, and lastly 9.4 Roles of Hormones during Pregnancy and Parturition. In this video, we are only going to look at 9.1 Sexual Reproduction in Flowering Plants. Okay, so these are the learning outcomes for 9.1 Sexual Reproduction in Flowering Plants. At the end of the lesson, students or you should be able to firstly, 9.1a, state and define the terminologies involved in gamete formation in flowering plants. So what are the terminologies? Okay, so these are the terminologies which are divided into male gamete and female gamete. Okay, I know it's a lot, but don't worry, we're going to look at each of these terms in the next few slides. Now let's look at 9.1b, explain the development of a pollen grain and formation of male gamete. The third learning outcome is 9.1c, explain the development of ovule, embryo sac, and formation of female gamete. And lastly, 9.1d, explain double fertilization in the formation of seed. So this is a diagram showing the reproductive structure of flowering plants. So if you can see in this diagram, there's a flower here. And if you look to the left side of the diagram, there's an anther in a black circle. Here. So inside the anther, there's a pollen sac or microsporangium. And inside the microsporangium, there's a microsporocyte. So this microsporocyte will undergo meiosis to produce 4 microspores, which are also called as tetrad. So each of these microspore will undergo mitosis to develop into a male gametophyte in pollen grain. So inside the pollen grain, there's a generative cell which will form 2 sperms and a tube cell which will form a pollen tube. So now let's look to the right side of the diagram. So this is an ovule. So an ovule is one of the female reproductive part in a flowering plant. Okay, so there's a megasporangium there, and inside the megasporangium, there's a megasporocyte. So this megasporocyte will undergo meiosis to produce 4 megaspores. However, only one surviving megaspore will undergo mitosis to develop into a female gametophyte or embryo sac, which contains 8 haploid nuclei, which are antipodal cells, polar nuclei, egg cell, and synergid cells. So we're going to look at each of these terms in detail in the next slide. So these are the terminologies involved in the formation of male gamete in flowering plants. So as you can see in this slide, there are 7 terms here. So the first one is microsporangium or pollen sac. Second is microsporocyte or microspore mother cell. Third is microspore. Fourth is tetrad. Fifth is pollen grain or male gametophyte. Sixth is generative cell. Seventh is tube cell. Okay, so let's look at the first term, which is microsporangium. So what is a microsporangium? Microsporangium is a reproductive structure in heterosporous plants that produces microspores. Okay, The second term is microsporocyte. Microsporocyte is a diploid cell in microsporangium that undergoes meiosis to produce 4 haploid microspores. Okay, so let's look at number 3. So what is a microspore? Microspore is a haploid spore in heterosporous plants that develops into a male gametophyte. Okay, so microspores are also called as tetrad. Okay, so when there are 4 microspores, it is called as tetrad. So what is a tetrad? Tetrad is a group of 4 haploid cells formed at the end of the second division of meiosis. Okay, so now let's look at the fifth term, which is pollen grain. So pollen grain is the immature male gametophyte of seed plants that produce sperm capable for fertilization. Okay, now let's look at number 6. Okay, so generative cell. Okay, so what is a generative cell? It is a cell in pollen grain that divides by mitosis to produce 2 male gametes or 2 sperms. And the last term is tube cell. So tube cell is a cell in pollen grain that produce pollen tube through which the sperm cells travel to reach the egg in the ovule. So now let's look at the terminologies involved in the formation of female gamete in flowering plants. So if you can see from this slide, there are 8 terms. So the first one is megasporangium. Second is megasporocyte or megaspore mother cell. Third is megaspore. Fourth is female gametophyte or embryo sac. Fifth is antipodal cell. Sixth is polar nuclei. 7 is egg cell. 8 is synergid cell. Now let's look at the first term, which is megasporangium. Megasporangium is a reproductive structure in heterosporous plants that produces megaspores. The second term is megasporocyte. It is a diploid cell within megasporangium that undergoes meiosis to produce 4 haploid megaspores. So the third term is megaspore. Megaspore is a haploid spore in heterosporous plants that develops into a female gametophyte. So the fourth term is embryo sac. So embryo sac is a female gametophyte generation produced after megaspore undergoes mitosis 3 times. Number 5 is antipodal cells. Antipodal cells are the 3 haploid cells in the mature embryo sac that are situated at the opposite end of micropyle. So it is situated at the chalaza end here, which is at the opposite end of the micropyle here. So the micropyle is the opening of the ovule. Now let's look at number 6, which is polar nuclei. Polar nuclei is the 2 haploid nuclei in the center of the embryo sac that fuse with the sperm during double fertilization to form a triploid endosperm nuclei. Number 7 is egg cell. It is the haploid mature female reproductive cell that is fertilized by a sperm to form a diploid zygote. The last term is synergid cell. It is the 2 nuclei in the embryo sac that are closely associated with egg cell to form the egg apparatus. Okay, so now let's look at 9.1b and 9.1c. Let's first look at the flower here. So there's anther and filament, which are also called as stamen. So this is the male reproductive part in a flowering plant. And here we have stigma, style, ovary, and ovule, which are also called as carpel. So this is the female reproductive part in a flowering plant. Now let's look at the upper part of the diagram. Here we have anther, and when we cut an anther, we can see that there's 4 pollen sacs. 1, 2, 3, 4, which are also called as microsporangia. So remember, when there's more than 1 pollen sac, it is called microsporangia. But when there's only 1 pollen sac, it is called microsporangium. So inside a pollen sac or inside a microsporangium, we can see here there's a lot of microsporocytes. Okay, so let's assume we take out 1 microsporocyte, 1 diploid microsporocyte. Okay, so this diploid microsporocyte will undergo meiosis to produce 4 haploid microspores. Okay, and each of these microspore will undergo mitosis to produce pollen grains. Okay, and inside the pollen grain, there's a generative cell which will produce two sperms, and a tube cell which will produce a pollen tube. Okay, so we've looked at 9.1b, how a pollen grain is developed and how male gamete is formed. Now let's look at 9.1c. Okay, let's look at the lower part of the diagram. So we have an ovule here, or the megasporangium. And inside the megasporangium, we have a diploid megasporocyte. So this diploid megasporocyte will undergo meiosis to produce 4 haploid megaspores. Okay, and then 3 of the megaspores, 1, 2, 3, will degenerate, leaving only 1 surviving megaspore. So this 1 surviving megaspore will undergo mitosis 3 times, okay, to produce an embryo sac with 8 haploid nuclei. Okay, so 4 of the nuclei, okay, so let's assume there's another nuclei here. 4 of the nuclei are located near the chalaza end. Okay, and another 4 of the nuclei are located near the micropyle. Okay, and then 1 nucleus from the chalaza end and 1 nucleus from the micropyle will move towards the center of the embryo sac to produce a single diploid nucleus. Okay, a single diploid nucleus, which is called as polar nuclei. Okay, so let's erase this one here. Okay, and then 3 of the nuclei will remain at the chalaza end to form antipodal cells. Right? And then another 3 nuclei will remain at the micropyle. Okay, so 1 nucleus will become the egg cell and another 2 nuclei will become synergids or synergid cells. Okay, so we've looked at 9.1c, how ovule and embryo sac are developed and how female gamete is formed. Now let's look at 9.1d, double fertilization. Okay, so before double fertilization can occur, there's pollination and germination. Okay, so what is pollination? Pollination is basically the transfer of pollen grains from the anther to the stigma. Okay, so there's a pollen grain here, and let's assume this pollen grain is carried by the wind. Okay, and then it lands on a stigma. Okay, so this pollen grain will then absorb water and germinate by producing a pollen tube. Okay, so this long pollen tube, okay, which it delivers sperms to the embryo sac. Okay, so deliver sperm. Okay, so now let's look at the next slide. Okay, so now we're going to look at 9.1d, double fertilization in the formation of seed. Okay, let's look at number 1 here. Okay, initially, the pollen tube consists of two haploid cells, the generative cell and the tube cell. Okay, the generative cell will then divide by mitosis to produce two haploid sperm cells. Okay, so one synergid degenerates when the pollen tube arrives. Okay, the sperm cells are then released from the pollen tube. Okay, one sperm cell will then fertilize the egg cell to produce a diploid zygote, and another sperm cell will combine with the polar nuclei to form a triploid endosperm nuclei. Okay, so that's it. So that's double fertilization. Okay, so what is double fertilization? It is the union of two sperm cells with different nuclei of the female gametophyte or embryo sac. Okay, so as we can see here, so one sperm cell will fertilize the egg cell to form a diploid zygote, and another sperm cell will fuse with the polar nuclei to form a triploid endosperm nuclei. Okay, so after double fertilization, the ovule develops into a seed, integuments become testa, and the ovary develops into a fruit, enclosing the seed. Okay, so I'm going to share with you a video about double fertilization in flowering plants.
Flowering plants undergo a unique reproductive process where there are two fertilization events. This double fertilization event occurs between the male reproductive organ, the male gametophyte, and the female reproductive organ, the female gametophyte. Before the fertilization event can occur, the ovule has to undergo some changes. At present, the ovule contains one reproductive cell, known as the megaspore or mother cell. This cell is diploid and undergoes meiosis, producing 4 haploid megaspores. In the majority of species, 3 of these megaspores degenerate, leaving only one surviving megaspore. This surviving megaspore expands and undergoes 3 rounds of mitosis to produce 8 haploid nuclei. As the nuclei have not developed any individual division, they initially share the same cytoplasm. This complete structure is known as the embryo sac. Within the established embryo sac, cell walls begin to form between most of the nuclei. 3 cells named antipodal cells form opposite the opening of the ovule, known as the micropyle. Another 3 cells form above the micropyle. 2 of these are synergids, and the other is the egg cell. This leaves 2 nuclei in the center of the ovule. These central nuclei remain together in one large cell. It is the egg cell and this central nucleate cell which will eventually become part of the double fertilization event. In order for the double fertilization event to occur, the male gametes, the sperm, must travel from the anther to the embryo sac within the female reproductive organ. The pollen grain contains two main cells. A cell named the tube cell makes up the bulk of the pollen grain, and the sperm cell, which at this stage is known as the generative cell. To reach the embryo sac, a pollen grain must land on the stigma. Once landed, it begins to germinate. The tube cell forms a long structure down the style and into the ovary. The generative cell travels behind the tube cell nucleus. Once near the ovary, it divides by mitosis to produce two haploid sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the two sperm cells fertilizes the egg cell. This produces a diploid zygote, which will become the embryo. The other sperm cell moves up and fuses with both of the central nuclei, forming a triploid cell. This unusual triploid cell develops into an endosperm and serves as the embryo's food supply during early development. It is only angiosperms, flowering plants, which have this double fertilization characteristic, where a diploid zygote and a triploid endosperm form. Gymnosperms, pines, tracheophytes, ferns, and non-tracheophytes, mosses, lack this double fertilization feature.
Okay, so that's it. Now it's pop quiz time. Okay, so before we end this lecture video, I'm going to ask you to scan the QR code, okay, here, and answer all of the questions, and good luck. Okay, thank you. Bye-bye.